Good afternoon, everybody. On behalf of myself, Mark Sondrop, and Wendy Spearman, my co-host for this afternoon's University of Cape Town Department of Medicine webinar, in conjunction with Project ECHO, University of New Mexico, that we're grateful to for extending their licensed platform to run these webinars. We welcome you again, and as you know, these webinars are now running every 2 weeks. Graham will fill you in more about how long we plan to do these till later. And before I hand over to Graham again, apart from Project ECHO, University of New Mexico, we're grateful to support from our colleagues in the Department of Medicine, as well as the Gastro Foundation, to enable these webinars. But without further ado, to hand over to Graham, your chair for the afternoon. Graham.
Right. Thanks very much, Mark and Wendy. So we're going to start off our usual format with an update on the epidemiology of COVID-19 in the Western Cape. And there are going to be two speakers who are going to provide brief inputs this afternoon, Andrew Bull, as well as Claire Draper from the Southwest substructure. And Claire's going to tell us about the recent outbreak among young people in that substructure. That will follow Andrew's presentation of an update of the numbers from the Western Cape in general. So firstly, over to you, Andrew.
Thanks, Graham, and greetings, everyone. Are you projecting for me, Mark? Great.
I am Andrew, you can proceed.
Yeah. So very briefly, this is a slide we've been showing for some weeks now, which integrates the cases in grey with the deaths in red, the officially reported ones, and those that we're estimating from our line list. Admissions in blue, admissions with public sector PUIs in the dotted blue, and the bulk oxygen supply in the public sector in green. Few things to note. You'll see at the bottom right a little uptick in cases in the grey, which we'll talk about a bit more. Hospitalization and mortality have been fairly stable in recent weeks, over a period of a couple of weeks, and especially in the private sector, there's been an ongoing level of admission which has come down from the peak, but is still sitting at about 40% of what it was at the peak. And we've confirmed with some of our private sector hospital group colleagues that this is real and not artifact. So they are still seeing a substantial ongoing burden. Yeah, and mortality has been low, but low stable. And in the excess mortality, which I won't show this week, there has been a small increase that is in excess of what we can explain from COVID, but it's off a low base compared to previous years. So it's still quite, with such small increased numbers, it's quite unstable, but something that we'll watch over the next couple of weeks. So on the slide, the top left is the testing numbers, and you'll see in the green, the public sector testing has been fairly stable for a period of time. An increase in private sector testing in the blue in the last couple of weeks. And in the graph below that, you'll see that the percentage positive in the private sector has midweek in a couple of weeks, it's hit 20%, running at about double what it is in the public sector. And that aligns with the increased case numbers. Look, moving across to the right, you'll see that the moving average in red of the number of cases has increased off our trough. So the national minister is has called this a resurgence. We definitely have seen this uptick, and it's been more. If you look at the green on the right, you'll see that the public sector cases have been fairly stable for a period of time, and that increase in cases is mostly coming from the private sector. And converting that into a reproductive number, is quite unstable with these small numbers. It was running at 1.3, and it's back. If you use the case numbers, which are very dependent on levels of testing, it's currently running at about 1. And just in terms of where we are with testing numbers relative to other countries, the Western Cape and South Africa are very similar. So on the vertical axis is the number of tests per million daily, and on the horizontal axis is the number of cases. And countries that are doing exceptionally well with suppressing the epidemic are on the top left corner. And the dotted lines show the positivity, and they're both on the both axes on the log scale. And countries who are serious about wanting to suppress in order to enable resumption of normal activities are trying to get as far across to the left as they can, and ideally top left. So it's sobering to see where we are, but we are in a company of many countries not dissimilar to ourselves. Looking at the cases geographically, this is the last 4 weeks by sub-district. Urban at the top, and you'll see the most especially southern and western, but also a little bit northern. We did last week have the 2 weeks before this, the last 2 weeks up until this week have quite substantial increases in numbers. Aligned with known clusters, and then this week the numbers are slightly down again. For the rural districts have been coming down consistently. They're on a different axis because Central Karoo was still coming off its first wave and was much higher. So you'll see that I think if we advance the slide, there'll just be a highlighting of the axes. Yeah. So you'll see that okay, we're on the next. Yeah, you'll see that the axis is slightly higher on the bottom graphs to accommodate the Central Karoo. Moving on to the next slide. This is the testing by age, and you'll see that the proportion that are younger in the orange are much lower when we were undergoing the larger numbers, but more recently, and especially in October, you'll see that there are many more under 20, and the number in the 20-29 group also increasing. It's interesting that the private sector testing is accommodating more younger people, and this might be more of the mildly symptomatic or asymptomatic testing in relation to returning to work or other activities. And public sector testing is probably more symptom driven. And you'll also see, looking by sub-district, and just in October, how the 2 sub-districts that you saw on the earlier graph, Western and Southern, have in October specifically got a much younger age distribution and also much higher numbers at those ages. So this really talks to the outbreak that Claire's going to talk about now. Yeah, so I think I'm going to leave it there, and I won't talk about admissions or deaths this week. But it also then gives me the opportunity to introduce Claire Draper, who's a family physician who has been supporting the Southern Western substructures since May with their outbreak response. And the substructure have really been phenomenal in the timeliness, completeness, and the fidelity of the outbreak response. And I think anybody on this call who has been diagnosed with COVID and lives in the jurisdiction of the Southern Western substructure probably has encountered the outbreak team who may well have called them after their diagnosis, such as the phenomenal completeness of the outbreak response that they lead. And so it's with pleasure that I hand over to Claire to just describe what they've been saying for the last few weeks. Thank you very much.
Thanks very much, Andrew. Maybe I could just start my video. and thanks for displaying my slides. That's great. So yes, I just want to talk a little bit about the young adult cluster that we identified in the southern and western substructure. For those of you not familiar with the substructure configurations of the metro, southern and western extends from Cape Point up to Atlantis, with southern being kind of Cape Point to the N2 and extending as far east as the N7, and western from the N2 up the coast up to Atlantis, again extending as far east as the N7. So it's the thin band of coastline. So quite a big area that we cover, but quite variance in terms of the kind of socioeconomic standing of those in our substructure. So as an outbreak response team, our job is to contact and call all of the cases that are diagnosed with COVID that live within our substructure. So we have a team of about 30 case managers that on a daily basis phone every single person in our substructure diagnosed with COVID, whether they're in the private or public sector, and just assess that they can isolate and quarantine their contacts, etc. So on the 9th, Friday the 9th of October, we received a call from one of our case managers to say that he'd just spoken to a positive case who reported that she'd been at a bar in the southern suburbs on Saturday the 3rd of October and had developed COVID thereafter, but also reported that many of the friends who were with her had also become sick after attending the same event. And in addition to this, I'm part of a private GP WhatsApp group that covers about 250 GPs, mostly working in the southern suburbs. And the WhatsApp group has started to support private GPs in clinical questions, but also particularly around COVID. And I noticed a number of comments on this group saying, has anyone seen that we just tested a lot more high school kids this week, or I've had a lot more positives than usual. And then there was mention of the same bar, that it seems like a lot of them had attended this bar over the previous weekend. And, you know, is anyone else picking this up? We were also made aware of a number of smaller social events and sports events that happened over the same weekend. And yeah, so it largely being due, it seems, to the kind of closing of out of the matrics year at school. So based on all of these factors that we heard about, we were concerned that we may be dealing with an outbreak, a cluster, perhaps even a super spreader event. And so we immediately notified environmental health for the city of Cape Town and asked them to investigate this particular location and to intervene. And we also notified law enforcement. I also contacted all the GPs who posted these messages and asked them to please directly email me with the details of any cases they were aware of within their practices that looked like they could fall within this sort of cluster. And the GPs themselves rapidly self-organized to support the schools involved, because a number of the cases were learners at school, many of them in matric. And many of the GPs have children in these schools, so they quickly self-organized, allocating themselves each to a school and to provide support and oversight for that school. And we also reported this potential outbreak to the provincial outbreak team, led by Prof. Hassan Mohammed, and he also then reported it to the Department of Education. Thanks, I can have the next slide. So we then spent the weekend collating a list of these cases that the GPs had forwarded to us, just based on what they had found out in their practices, and cases that were either linked to schools or sports events or this bar attendance over the weekend of the 1st to the 4th of October. We also, in our COVID case management, we utilized the provincial COVID database called Single Patient Viewer. And this is a database on which every single positive COVID case appears and is sorted by those cases that live within our area. So we were able to then cross-check all the cases we'd been alerted about and to just see, had all these cases been reported, were we aware of them, and to develop a kind of a mini database of this potential cluster. Then on Monday, when usually every day, any new case that appears on our line list, we allocate out to our various different case management teams throughout our substructure. We have about 30 case managers in various teams. But for Monday, we deliberately allocated all the young adults, the 13 to anyone age between 13 and 23, we allocated all to one team. And we instructed the team to please ask about whether these were scholars, they attended school, and whether they had attended this particular bar. To help us understand this outbreak better. We also informed all our case managers throughout the whole substructure and alerted them to please, if any case they were phoning, to please ask particularly about this cluster. We use an MS form reporting tool in order to receive data back from our case managers. So every case that they call, they complete a form which just gives us some details about the case, whether they were able to contact them, the number of contacts. And I added an additional question to this form to ask the case managers whether they felt that this case was part of the cluster. So we did that just before the week started. So this has all kind of emerged in the last 10 days. So obviously we're still finalizing what we've seen, but so far we have identified 79 new COVID cases within the southern suburbs across 9 different schools since the beginning of October. And 43 of these learners are currently in grade 12. We've also identified 109 cases that are linked in some way to possible super spreader events that happened over the weekend of Thursday the 1st to Saturday the 3rd of October. 57 of these cases are learners, and at least 11 cases identified in this cluster attended the bar on more than one night over this weekend. So while it encompasses 3 nights, perhaps not just one event, it seems that there was attendance on more than one night, which could perhaps have promoted spread. So just to give you an idea of how things have changed in our substructure, 2 weeks previously, in the sort of middle week of September, we had a total of 287 cases, COVID cases in our substructure, of which 40 of those were between the ages of 15 and 25. So roughly 14% of our caseload were young adults. And in the middle week of October, at the height of the numbers that came through, we had 542 cases, of which 180 were between the age of 15 and 25, so 33% of our case load. So you can see a clear swing towards a much younger age group and an almost doubling of our case numbers in a 2-week period. So just a caveat in all that I'm saying, while we've done our very best to verify the links between all of these cases and their contacts in this cluster, based on the histories given to us by our case managers and by local GPs, it's obviously impossible with 100% accuracy to determine the links of this. And to this end, we have engaged with medical virology at Tygerberg to assist us with viral sequencing. And we're hoping that this will give us a more accurate idea of the pattern of spread and the virulence of the strain, so that we can more accurately map this cluster. So this is just a graph that has been produced by Andrew and his team, and just shows that what the cluster looks like. Andrew did show something very similar in his presentation, but you can just see the clear spike in our numbers so the green is the southern substructure, and of those cases aged between 10 and 19, you can see a clear spike there over the period, the last day of September to kind of the middle of October. And a similar but less pronounced spike is there as well in the age groups between 20 and 29 years. So that's evident to see as well. Thanks. Then this is just from our provincial dashboard that we can use to track cases and age groups and death rates, etc. And I've just taken a screenshot of the provincial dashboard yesterday. And oh dear, my slides seem to have disappeared. I'm not sure if they've just disappeared for me or if they've disappeared for everyone.
That disappeared for everyone, Claire. I think okay. Mark's trying to get them back. Yeah.
Okay, perfect. No, that's fine. No problem. Yeah, it was just the provincial dashboard there. That's great. Thanks. So yeah, you can just see that basically what I've done is I've just selected out for the southern substructure, and then I've just selected out for the age group 6 to 20, and it just gives a very clear indication. As you can see, the line was tracking kind of almost plateauing until the beginning of October, and then there's a sharp uptick particularly in that age group, and you can see it represented in actual cases in the white down below. Looks like, thankfully, it's slowing down a bit, but yeah, it's just very obvious on that graph to see. Thanks. You can have the next slide. So this is just a graph showing the number of cases that exist in our southern suburb schools since the beginning of October. I've obviously taken out the names of the schools for confidentiality, But we had, the school with the most cases had, since the beginning of October, has had 18 cases. Other schools with 14, 11, less schools with less high numbers having just 4 cases. But in total, we've had 79 cases of learners and school employees. These cases don't exactly dovetail with our cluster, but our concern is that there may have been spread within the schools based on those learners who did attend these social events. So the next slide. So yeah, this is just to indicate how many of these learners were grade 12 learners. And as you can see, in most of the schools, bar perhaps school 3 and school 4, most of the cases that were that did develop COVID were matrics, which is obviously concerning at this time of the year when matrics are about to start writing exams. So this was a big concern for us, especially to just address that and make sure that schools were adequately supported and we could contain spread within these learners. So then this is all the cases that are linked to our possible super spreader event. So as you can see, a number of them occurred within schools, but there was also quite a significant number that happened in the post-school age group, either university students, young working adults. And so there were a significant number that added to this cluster that were not school attendees. So yeah, at the moment, the total number that we've identified that either attended the event directly or were close contact, or at times a close contact of a close contact who's been now infected the second and third wave, is 109 cases at this point. And then this is just indicates the spread of attendance of the super spreader event. So the majority of the cases attended this particular location on Saturday, the 3rd of October, but there were also a significant number that attended on Thursday, the 1st of October. Not nearly as many attended on the Friday. Also, so far we've identified 23 close contacts that have developed COVID as a result of contact with someone who attended one of these parties. And for 23 of the cases, we established that they did attend on one of these nights, but it's just unclear which night it was. Just of note to say that at least 11 cases and possibly more did attend this particular venue on more than one occasion. So at least on at least 2 nights, mostly on the Thursday and the Saturday night. So in terms of our outcomes, this is obviously an ongoing investigation, and we continue to provide information to environmental health, to the South African Police, to the Department of Education, and to the Provincial Outbreak Response Team. We've also attempted to raise awareness through statements to the media, local school engagement, and our communications department has awareness campaigns planned targeting young adults for the near future. We also have ongoing monitoring and contact tracing that is occurring by the GPs in our area who are linked to the local schools that are involved in this outbreak. We are also just keeping a close eye on boarding houses, as these are possible areas for subclusters to develop. Our case managers also continue to phone any new cases that arrive on a daily basis. And we did right at the beginning alert Uber based on the assumption that they may well have transported cases home from a night out, and that their staff may be at risk. So they have been alerted. And then in terms of our learnings, so I think as a team, we just learned the value of technology to allow for rapid action. So first of all, WhatsApp group communication and just being able to pick up on trends that way from a much broader audience. And also just the use of SPV in order to help us keep track of these cases and locate them. Also just saw the value of public-private collaboration. So a lot of these, since this outbreak was largely in the private sector, it was very useful to have connections with private GPs on the ground and get their input and their sense of what was happening as the first responders. We've also learned the value of and the role that viral sequencing can play and the cluster mapping. And this is an ongoing project for us to just try and understand this outbreak better. And I think lastly, just the value of a primary care provider's involvement of schools. And we've really seen this with our GP colleagues in private, who've really gone above and beyond to support schools and try and assist wherever they can to make sure that outbreaks are contained and that learners are kept safe and that protocols are put in place. So that's all I have to say. Thanks very much for having me.
Right. Thanks very much, Andrew, and to Claire in particular for that detailed insight into this outbreak. And congratulations to you and your colleagues for the rigor with which you undertook this outbreak investigation, as well as the way in which it was communicated to the public. And I think that's had an important role in controlling this particular outbreak, hopefully. So we're going to move on to our main talk for this afternoon. And this afternoon, we're taking a step slightly sideways from a direct focus on COVID-19 to hear about research on TB aerosol transmission. And as you're all aware, there's been a major debate internationally as to the relative contribution of large droplets versus aerosolized particles in the transmission of SARS-CoV-2. And the thinking behind today's session is really that an understanding of the aerosol transmission of TB that we're going to hear about may help to inform our discussions and understanding around potential SARS-CoV-2 aerosol transmission. Now, it's a great pleasure to introduce our speaker for today, Robin Wood. Robin is an Emeritus Professor in the Department of Medicine at UCT, as well as CEO of the Desmond Tutu Health Foundation and Director of the Desmond Tutu HIV Centre. And Robin has been a leader in HIV-TB research in South Africa since the 1990s. His recent focus has been on the aerobiology of TB as principal investigator leading a scientific research group of the MRC flagship program investigating the aerobiology of TB transmission. And in his presentation today, he'll discuss the work that his group has undertaken, and that'll lead into a discussion about the implications of the findings for understanding transmission of other respiratory pathogens, including SARS-CoV-2. So thanks very much, Robin, and over to you.
Okay, thank you for the opportunity to talk about some of our work. This is our research center down at the Masiphumelele site. It's been open for almost 2 years now. Next slide, please. Did you get that? Next slide. I don't have control. Do you?
We're seeing the next slide, Robin.
Oh, you're seeing the next slide, but I'm not. Okay, wonder why that is. I'm still seeing the first slide.
Okay.
Okay, so I'll have to do from what I remember. Essentially, I'd just like to very briefly go over the major advances in aerosol science over the last few years. I'm still on slide 1, by the way, on my picture.
We can't fix that, Mark. Is it possible to move it?
Okay, so this was going back 100 years. This is some work from Chaussee, and essentially he took tuberculotics and got them to cough into this box, and well hidden within that box, and probably this is the first time to show that airborne transmission could take place. And essentially, he found about 38% of the patients were able to infect the guinea pigs. Next slide, please. So we can move forward another 50 years to the sort of seminal work of Riley and his collaborators. And at that time, there was still a lot of debate about whether TB was aerosol borne or whether it was fomites. And he really put this question to bed by taking the air from a TB ward and passing it through the air conditioning system to the guinea pigs. But he found that patients starting on treatment couldn't infect guinea pigs. And in fact, he found about 13% of patients were able to infect the guinea pigs. And they did some calculations. The infectious units were incredibly rare. You'll see down at the bottom, 1 infectious unit for almost 400 cubic meters. So it gave everybody the idea that we were looking for a needle in a haystack. Next slide, please. So we go forward another 50 years to the work of Fennelly more recently. And essentially he developed this cough box, which is really much the same as the Chaussee experiment in 1916, except he replaced the guinea pigs with Anderson impactors, which are devices for impacting aerosols onto solid media and trying to grow them. And in his initial experiment, and this has been repeated several times subsequently, about less than a third of the patients were infectious. Next slide, please. So if we put that together, a sort of 100 years of aerosol science showed a small proportion of sputum positive individuals, and it's still sputum-based diagnostics were the thing that everybody relied on. Next slide, please. So Graham mentioned the flagship project, UCT flagship project, and it was a very interesting multidisciplinary team. And because of the idea that these TB organisms were so incredibly rare, 1 per 400 meters, we called the project Hunting the Boson. But this is probably the first picture of an airborne TB organism, and this is an impaction onto an aluminium slide, and it's a scanning electron microscope picture that was done by Jeremy Woodward from Upper Campus. So we were quite encouraged that we could with defined organisms. We then set about, can we improve the collection systems, and can we improve the detection systems and see what we can find in aerosols. Next slide, please. So this is, there's a big delay, but from what I remember, the slide in the top left-hand corner, you see one of these very fast pictures of, it's said to be a cough, but I believe this is probably a sneeze. And what you see here is a series of trajectories of different particle sizes, the larger particles falling out very fast, and the smaller particles being contained in a moist, warm cloud, which moves away from the patient and actually moves upwards. So we try to think, where are these particles coming from? So the 3 parts of the respiratory tract, we start at the peripheral lung. Interestingly, the particles are produced during inspiration as closed airways pop open. They're surfactant, which is basically a type of detergent, and there is very little flow here. Most of the air movement is by diffusion. So we think that just opening and closing the peripheral airways produces particles. Obviously, it's been postulated that fast flow through the larger airways might also create shear forces and create particles. And then lastly, we can add to that vibration of the upper airways, sneezes, particularly loud speech, etc. Next slide, please. So the problem with collecting this is a cough actually comes out at about 40 meters per second. And it's very difficult to capture that because the air is incompressible and basically bounces off surfaces. So we thought about how we would do this, and what we came to the conclusion is that we would have to collect the expired air in a very fast stream of computational fluid dynamic modeling done at Upper Campus by the physicists up there and Arnaud Milan's team, so that we could collect efficiently a cough, and that we would then take that output in particles, monitor the carbon dioxide levels so that we could see the volume of the cough or other respiratory maneuvers. We'd look at the particle sizes that are coming out, and that we would collect the particles into liquid in order to detect MTB. Next slide, please. This is a picture of our sampling chamber. Patients go into that wearing a Tyvek suit to decrease any background contamination of particles, and that allows us to collect direct particles that are coming straight out of somebody's exhaled breath, and also contaminated space to the environment and stay in the environment. Next slide. So this slide is a little bit complex, but just to concentrate first of all on the top left-hand portion, which is a direct collection showing you what we find. This is 10 coughs. You see in the blue at the bottom the carbon dioxide levels, which rapidly increase with the explosion of the cough and is then collected. And you can see how reproducible they are within an individual. And in the upper part of the graph, you see the particle sizes that people are creating. And the first thing you notice is that the green line, which is large particles above 5 microns, this is a log scale, are incredibly rare and don't seem to be particularly related to the respiratory 5 to 5 microns in size. The colored particles at the upper portion of the graph are related to the maneuver. So what it's showing us is that in this individual and in other individuals we found subsequently, the particle sizes produced during a cough are between 0.5 and 5 microns, with very, very few larger particles. I think this might have relevance for the COVID talk. So because of the sensitivity of this, we were able to, in fact, compare different maneuvers. So the in panel B, you see 5 deep breaths, which are creating these particles in the peripheral of the lung. You can see tidal breathing, which was interrupted by 1 cough. You can see there, and then you can see a series of coughs. And the particle sizes, again, are predominantly between 0.5 and 5. If we move over to panel C, what this is illustrating is that when you collect particles directly, and that's the time of flight of less than 2.5 seconds, you collect many more particles than if you wait for the particles to adapt to the environment. We estimate that the time of flight for there is about 2.5 minutes. So you can see again that the particle sizes are between 0.5 and 5 microns. Very few larger particles are produced, but that this difference is due to particle loss and particle change in size as the relative humidity decreases from the 100% humidity in the individual. Next graph, next slide, please. So this is just to look at on the right-hand side, the comparison of volumes rather than numbers of particles, because as you remember, volume of a sphere is 4/3 pi r cubed. So the larger particles have much higher volume than the particles. But what I'm really using this slide for is to look at the left-hand side. And there you can see that the volume of bioaerosol we're collecting is pretty similar in each of the 3 maneuvers. And when we put those 3 maneuvers together, we're probably collecting in our chamber between 1.4 nanoliters and 3.4 nanoliters. So we've got a handle of how much air, how much time, and what volume of bioaerosol we're collecting. Next slide, please. So we move from the efficiency of collection to the detection of TB. And what we started using was this new probe that was developed a couple of years ago, published in Science from the Bertozzi group in Stanford. And what she did is got a sugar trehalose analog and linked it to a fluorophore, which fluoresces. And this is metabolized by organisms that have an antigen 85 complex. And then it's metabolized and incorporated into the growing capsule. Next slide, please. So some of the characteristics of this are that, first of all, the organisms that have an 85 homolog are relatively few, and we show the major ones there. The one we were more concerned about, besides the tuberculosis, was the Carini bacteria, which can be contaminants of the upper airways. But on the right-hand side, you see the characteristics of a salvadochromic probe, and that is that the fluorescence is increased massively, this 700-fold, in a non-aqueous environment. So this is in the fatty layer, waxy layer of the outer high fluorescence, but in the aqueous solution, you'll get very little fluorescence. And this means that you'll get a much better signal to noise ratio. Next slide, please. So what we're showing here is a Mycobacterium sphancmatis, and you can see on the left-hand side the way the stain, the DMN tray, is taken up. That's the green fluorescence. You can see the growing areas, ends that of the organisms light up. The red is a chromosome marker with M cherry. And you can see that we can actually measure the fluorescence along each organism. You see that at the top right. So you can see the fluorescence is concentrating at each pole, but you notice that when organisms are about to divide, that you start seeing some fluorescence organisms about to split. We can look at the length, the shape and size of these organisms as well. So move on to the next slide, please. These are the way that we identify the organisms. We spread the bioaerosol over these nano wells. So each one is about a nanoliter in size. And there's 1,000 to 2,000 of these on a slide. And again, the idea is that we dilute the noise widely, but an individual organism would go into one of these wells, can be then observed. It can be given an address so that we can go back to it and see how it changes over time. Next slide, please. So these are some examples of staining. Concentrate on panel B. You can see these different characteristics of the organisms. They can be sort of segmented, round or more oval. In C, you can see occasionally we come across clumps of these organisms, and at the lower two panels, you can see that we can compare the sizes of these with a normal laboratory strain of TB, and essentially most of ours seem to be a little bit shorter than the laboratory, but a much wider spectrum of widths of organism in the second panel. Next slide, please. So here we see the results of, we can see a marked variety of phenotypes of organisms. So here you're seeing this beaded appearance in A. You can have curved organisms. You have these that take up the stain relatively faintly. We have some which are acutely angled. We have some which are taking up the dye at the poles. You can have a diffuse, oval, and some forms are filamentous. So we're seeing a very marked phenotypic variety of TB organisms. Next slide, please. If we look at the numbers of organisms we're collecting, and here we're comparing the, we're adding the results from bronchial burst, tidal breathing, and cough, you can see that we have probably 1 to 2 variability, and a lot of variability is which mover gives us the organisms in large number, but this is a continuous distribution of organisms collected from 30 TB suspects. And on the right-hand side, you can see something which is quite interesting, that the deep breath, the force vital capacity bronchial burst maneuver produces identical number of organisms as a cough. So we believe that this indicates that these are being produced in those peripheral opening up of the airways. Next slide, please. So having increased the sensitivity of detection, the obvious answer is, are we actually finding Mycobacterial tuberculosis, or could this be some epiphenomenon? So this is just highlighting the organisms that have a antigen 85 homologue. They're all members of the Actinobacteria. It's the spectrum of organisms that we are going to see taking up this dye. Next slide, please. So if we compare what we're seeing with the known laboratory culture states of starved, stationary, and log phase, and you can see the fluorescent patterning in the middle, you can see that we are finding things that seem to be parallel to those, very similar to those. Next slide, please. So in order to convince skeptics, we took a second sample from all the patients that had a positive DMN tray, and then we used a conventional stain, Auramine O, which is known to be a mycobacterial specific stain because it's taken up in the mycolic acid layer of the 23 of our 24 positives were also positive for Auramine O. Next slide, please. So that really tells us that it has to be one of the organisms with an antigen 85. It has to be an organism that is a mycobacterium. So what we then did is took a second sample from the patients again, and we ran a PCR assay looking for the DNA sequence of RD9, which is a marker specific for MTB. You see on the left-hand side that other mycobacteria in the Bovis and Africanum spectrum have lost the RD9. It's something that was lost fairly early on in that differentiation. And here we had 100% of our DMN tray were positive for RD9. Next slide, please. We took the bioaerosol and did some mass spectrometry analysis, extracted lipids, and looked at lipids in the positive ion mode and the negative ion mode. And what you can see here is that it seems to have quite good discriminatory information. This is principal component analysis between controls, non-TB, and TB patients. This is due to a combination of human lipids, which are in the principal component analysis, and mycobacterial lipids. And we have managed to identify some specific mycobacterial lipids, such as the PIMs, PIM1s, etc. So again, this shows that the bioaerosol contains mycobacterial lipids. Next slide, please. So finally, this is some preliminary work, small numbers, but statistically significant. What we did is we repeated aerosol sampling at 2 weeks and 2 months, and we could show that there was a significant decline with treatment. So we think that if you put this evidence together, that it does point to what we are identifying are indeed Mycobacterial tuberculosis. Next slide, please. So if we go back to the aerosol detection, we're now finding positive organisms in 100% of the sputum positives and a very large proportion of the sputum negative cases. So we think that by improving the efficiency of collection together with very sensitive detection methods, that we are moving into an era where aerosols may well be quite an important research tool for the future. And we also think we're quite excited about the prospects of single cell mycobacteriology. Next slide, please. So in conclusion, we think that respiratory activities predominantly produce respiratory particles in the range of 0.5 to 5. I suspect that speech, may add larger particles from the very upper airway, the oropharyngeal airways, but the lung production is predominantly in this range. We think that we've extended the bacteriological identification of mycobacteria to a much wider spectrum than previously with sputum positive diagnostic. We think that we were a little bit surprised that we could identify organisms up to 2 months into therapy. We haven't looked longer than that, but this certainly gives us the opportunity to monitor the effects of and late specific TB therapy. And the interesting thing about the DMN-Tray probe is it's non-destructive. It's incorporated into the wall of the organism, but it doesn't kill it. It still remains alive. And certainly when we go back and look at the address of these organisms up to 3 weeks later, we can see the fluorescent particles. So we're quite interested in looking at developing clusters derived from single organisms so that we could, in the future, move on to whole genome sequencing based on single organisms. And I think this has some implications for TB vaccine and perhaps treatment protocols like INH, that it seems as though if a larger proportion of the population are living with viable organisms and in balance with their immune system, then obviously this has major implications for vaccine of the MTB in order to stimulate immunity. And similarly, INH is likely to be perturbing that relationship. Next slide, please. So just to give you an idea of the future studies that we're planning, we think that the single cell observation of growth, reproductive potential of the different phenotypes is going to be really interesting. We want to move on to in situ growth of the organisms and move on to whole genome sequencing. And from an epidemiological point of view, we want to see what the association between identification of aerosol organisms is with standard immunological markers, such as transcriptomics and gamma interferon assays, etc. And it raises the question of the disease and when do they clear it, and is the clearance at the end of therapy complete, and when are people acquiring primary and secondary acquisition of these organisms during childhood and adolescence, and particularly after successful treatment. Next slide, please. So a lot of data here. It's a multidisciplinary team. You can see in my Aerosol Research Center, our laboratory and clinical staff. Then we're collaborating very closely with Valerie and Digby's unit at the IDM Microbiology. We're grateful for the supply of the probe from Stanford University and our colleagues there, and Zateo Tech, which are interesting group of people who are heavily involved in USA. Certainly share a lot of the particle science and aerosol science data with us. And lastly, to our funders, which are the Bill and Melinda, SA, MRC, and the National Institutes of Health. So thank you very much for your attention.
Right. Thanks very much, Robin, for that fascinating overview of the work that you and your team have been doing on the aerobiology of TB. So we're going to move on to the panel discussion, and just to briefly introduce the panel members. We've got Rosie Burton. Rosie is an ID specialist. She's working now with Médecins Sans Frontières. Ayanda Trevor Mnguni. Trevor is a pulmonologist and head of medicine at Khayelitsha Hospital. And then Digby Warner, who is a TB scientist based at the IDM at UCT, and also working with Robin on this program of research. But Robin, could I just ask the first question and relate some of the work that you've been doing to SARS-CoV-2? Obviously, one of the phenomena that's been observed with SARS-CoV-2 is that a substantial proportion of the transmission occurs in patients before they are symptomatic, so before they're coughing and sneezing. And I'm just interested whether you've, any of your work can shed light on what are the sizes of the particles that are being produced in patients that are, producing particles during normal speech or breathing. Are those in the range of the larger particles or aerosolized particles?
I think that the aerosol scientists believe that it's in the size from 0.5 to 5. What I showed you with the graph with the direct versus the indirect shows that if you were in the range of the warm, moist plume, then you are exposed to many more particles than the aged particles. And I think people have confused that. Ballistic and hitting people.
Sorry, we lost you when you said people have confused that?
They've confused the proximity. We know that the disease seems to be related to closeness, but if you're within that warm, moist plume, then even the small particles are going to survive, and there are many more of them. Whereas if you're distant, then you have the dilution factor of the third power of the distance. I think that infectious particles are mainly from in this range. People don't spit at each other generally. So I think that people have by inference said, oh, well, this must be large particles. In our experience, people don't produce large particles, but you do see them when someone's speaking very loudly or or singing, etc. So I think size are produced by every activity. And I think it's because they're actually produced during inspiration, not expiration.
But with the slide that you showed, within a minute or two, there was a very substantial decrease. So obviously that's the time of highest infectivity in close proximity to people.
Yes. Yeah.
So if I can move on to the panel then and just ask Rosie if you've got one or two comments or questions that you have for Rob.
Thank you, and thank you for a fascinating talk. I'm thinking about this just from the point of view of being a clinician, and particularly working in very low resource settings where infection control is a huge challenge. And one question that I often have in practice is, well, when can we actually make a decision that a patient with TB is not infectious? And this is both, before diagnosis. So, I mean, if a patient has obvious pulmonary TB and is coughing, that's one thing. A lot of the patients with advanced HIV aren't coughing, they're not producing sputum. Some may have more a clinical picture of meningitis rather than pulmonary TB. So one question is, well, can we decide that those patients are not infectious? It obviously worries me that they are. But if somebody that we clinically decide has TB but doesn't have any pulmonary manifestations. And the second question is really is really the other side of this. When are patients with drug-sensitive TB started on TB treatment? When can we assume they're no longer infectious? And we always say 14 days. I've always hoped it was a lot less. Particularly we want to send patients home. We want to send sometimes to other institutions. But I see from your work that at 2 weeks there's still a substantial proportion of a substantial number of organisms you were detecting, and even at 2 months. Are those live organisms, do you think? And therefore, should we, are patients actually infectious for longer than we think? So, that particularly is one of the things I picked up in your talk.
Yeah, so yes, they are alive. Well, they're metabolically active, but whether they're infectious is the crux. And certainly the work of Riley with his guinea pig infections showed that very soon after therapy was initiated, that they could no longer infect the guinea pigs. So we have this interesting thing about these phenotypes. Obviously, this organism is in balance with the immune system, because if you were exhaling infective organisms, then you should spread them throughout the rest of your body, and you don't. So of smear positive cases probably explains about 30% of the transmission. That's most of the studies in high prevalence areas. You can establish some sort of chain of transmission in about 30%. So that would suggest that there's another 60%. So what I think is there's a large proportion of the population that are exhaling organisms, and that their infectivity is very inefficient. It's only when you're in the circumstances such as crowding, poor ventilation, Pollsmoor prison, etc., that under those circumstances, the transmission can be more significant. So I think there's a bit of truth in each of these ideas, but I think the question about MDR, I do not know that any of the data I've got really would answer that. But there's a difference between being metabolically active with infection. I think that the argument from Riley, etc., that you just needed a single organism to establish infection doesn't seem to fit with the data that we're showing. But we don't know whether it's those clusters that are infectious, whether it's a quantity picture. But essentially, if 80% of the South African population is infected, 1% get TB, 50% of those have smear positive. They're driving a lot of the 30% of the transmission. But one of the things that's always puzzled me is the acquisition of infection by children is very high during adolescence. And when I track smear positive TB cases and give them GPS monitors, they never meet the children. So the children are obviously getting infected from another source. And that's really what I'm trying to establish what that could be. And I think this work would suggest that's possible.
Right. Thanks, Robin and Rosie. So Trevor, any questions or comments that you have?
Yeah, thanks. Thanks, Prof, for that fascinating talk. I think I've just got two questions. And for me, the first question evades to the sort of negative pressure rooms that we have for patients with TB. And ideally, when they're infectious, ideally, we'd like them to have them in those type of settings. But in limited resource settings, you usually don't have access to negative pressure rooms. And it's previously been thought that you could put a sort of a smear positive patient at the end of the ward with windows open. Now, with regards to the air changes per hour, is that adequate enough to sort of decrease aerosol transmission? That's the first case relating to TB. And then the second case, just relating to your work and maybe trying to see how we can sort of link it with COVID and aerosol transmission with regards of COVID, is can you give a comment on the transmission in confined spaces of COVID, especially in say air travel, cars, and maybe buildings? Thank you.
Yeah, so air changes per hour really come down to the per person ventilation. We have done quite a lot of work on carbon dioxide levels. And my feeling is that if you want to control airborne diseases, you need to maintain the carbon dioxide level at less than 1,000 parts per million above ambient. At 4,000 means that every breath you're taking, 10% of it has been breathed out by somebody else. And certainly when we measure in schools and in certain environments, we get those sort of levels. Whereas I think there should be an occupational level of at least a minimum 1,000 parts per million. Then the second part of your question is the COVID. I think undoubtedly there is these and with distance. And I think the argument about aerosol versus particles is completely fictitious and is created by people who don't understand the particles. I think the key thing about the size of the particles is where they potentially can infect. So 5 micron particles will tend to get collected in the upper airways, whereas 1 to 2 microns will settle in the peripheral lung. So for COVID, we need to know where is the site of infection, and the very small particles tend to go into the alveoli and come out again. Obviously for TB, you need to have a particle big enough to contain a 2 micron particle. So that's 1 to 2, which we think is the size. So my feeling is, and I did get asked to come into the MAC on distribution of the, but it's become so political with people and aerosol, which is nonsense. And I think it's proximity, it's that plume that's being jetted off. So it doesn't mean that I don't believe a mask will not interfere with that plume and redirect it away from people. But these particles are 0.5 to 5 microns, and a proportion of them will survive for a significant time in a room. But obviously they're getting diluted by the airspace, whereas when you're close to someone, they can be directed straight at you. So I think it's a sort of nonsense to start arguing about the particles. Ballistic particles have to be 50 microns in order to sort of hit somebody in the eye, etc. And I think those are incredibly rare.
Okay, thanks, Trevor. And so Digby, to bring you in, you've worked with Robin on this project. Any comments or questions?
Thanks, Graham, and thanks, Robin, for the presentation. It's always so nice to see. I think really pioneering work over the last 6 or 7 years pulled into a 35-minute talk in such a concise way. I think one of the questions that I had going into this, and which has been touched on in Rosie's question at the outset, was as mycobacteriologists, we've generally taught students that this is an obligate pathogen whose life cycle depends on transmission to naive hosts. There's no environmental reservoir, and so to maintain a position in the host population, transmission is an essential component of the life cycle. And following on from that, we've basically operated, I think, in a paradigm that either very few organisms are produced which are very efficient at infecting, or perhaps much larger numbers of organisms are being produced which might not be that efficient at infecting. And you mentioned there the hunting the boson work. I just wonder if you may just want to amplify or augment what you said earlier. I mean, do you have a sense now where you sit on that spectrum? Is this do we have any sense of how efficient an infective agent MTB is?
Well, I think one of the things that I struggle with is what is actually happening in the periphery of the lung to maintain. So obviously there has to be a cycle within the individual, but somehow or other that balance with the immune system maintains it at a low level, so it doesn't produce disease. So it raises the question of what is disease? If you're living with the organism, the majority of the population, which is what we suspect, we know that if you give somebody a TNF block, they get diabetes, that balance gets rearranged. So that's the intrinsic thing of, interesting what's happening within the individuals and why if people are breathing these organisms out, and we're sort of calculating about 1 per liter is the range that we're talking about, they should be contained within the dead space and breathed into other parts of the lung. And yet people don't get disease. So not all of these phenotypes are able to infect within the individual. So now you're asking the question, well, what happens when it goes to somebody else? And I think there's too many questions there. We need to look at these phenotypes and make assumptions. Is the speed at which they grow, their growth potential, is that a marker of certain phenotypes? But the other thing that we've sort of struggled with is, why are there so many phenotypes? Why, when you grow in culture, they're all the same name? Here we're seeing this incredible diversity, and I think you're the one that's told me we've lived in balance for 70,000 years with this organism. Why is the organism producing this diversity? There has to be some reason for that, and I think that we are just going to be beginning to understand some of the complexity, and I think these new tools that we've got, it's quite an exciting time to be looking at TB, but I can't really answer your question at the moment.
It was deliberately provocative. Maybe if there's time for one more. The other thing
one more question, Digby, yeah.
Yeah, sorry, I was gonna also wondering, I mean, the literature suggests that HIV positive individuals are poor TB transmitters. Is that something that is evident in the data emerging from this work? And maybe just to bring it back to the COVID setting of this webinar, how do we know anything about COVID transmission against the background of HIV positivity?
I don't know. I know there's an association with disease, and therefore one would imagine that the acquisition of disease is related to HIV, but I don't think there's any data really that I'm aware of the ability to donate COVID to other people. But I think, I mean, Graham's work, this interaction and why we don't get disease all the time is really fascinating. So HIV in TB changes that balance between the immune system and the organism to allow more very severe HIV seems as though it decreases the ability to contain it within one body compartment so it can spread elsewhere. And then Graham's work is you give people something that makes their immune system better and then they get sick. So I think there's a lot of complexity and I struggle. What is it? Is disease caused just by numerical numbers of organisms? Or is there something else? Is it related to the phenotypes, etc.? And I think we need to look at that. As far as COVID, I think the parallel is that we can explain 30% of TB transmission by the highly infected sick patients. 60% we can't. And I think COVID is coming out the same way, that asymptomatic carriage is going to, at a population level, be quite significant, even though at an individual level, it may not be as apparent.
Thanks, Robin. So I'm going to hand back to Mark. I think Mark's got one question.
Thanks, Graham. Robin, Mark here. The fact that I was once your medical registrar emboldens me to ask you a question or your opinion. Given the mass sort of mask wearing that's been happening in our society over the last few months, do you think it will have any impact upon TB transmission in 2020?
I would think it would have an impact on the 30% of the high efficiency, which is the sort of transmission that can take place in westernized societies, whereas it's unlikely to make a difference from the low grade. So essentially 99% of the people infected with TB have a very low infectivity, but manage to eventually, over a long period of time, infect the majority of the South African population. So that efficient transmission from the sort of smear positives certainly will have an impact. Whether it will have an impact at this other level, I suspect not. But I don't think so.
Thanks, Rob.
Okay. So thanks to finish up, to say thanks to Robin, to Claire and Andrew for 3 excellent presentations. We really had a great focus on transmission of COVID and TB this afternoon, and to our 3 panel members as well. And then just in closing, to say that our plan for the rest of this year is to have 3 more webinars. The next one will be on the 11th of November, and that will be a talk from Barry Bloom from the Harvard School of Public Health on the public health perspective on COVID-19, really from a global perspective. And then a further webinar on the 18th of November focusing on long COVID, and then the final one on the 2nd of December covering a summary of the evidence regarding the therapy of COVID-19. So the next one will be in 3 weeks' time on the 11th of November, and we look forward to having you back on the webinar at that stage. And just to say thanks again to everybody who participated today for a very successful webinar. Thank you.
Thank you.